What's the next stage for developing your research with a view to producing treatments for Parkinson's disease?
Well, I think in the short term, our job is really to go out there and communicate to the clinicians and neurosurgeons at the coalface of Parkinson's disease and its treatment, to really think again about this structure, the external globus pallidus. So this particular brain structure has fallen out of favour as a target for surgical intervention in Parkinson's disease, particularly because of a large literature suggesting that if you target that structure, it would be bad news for the treatment of Parkinson's. But our work really quite strongly suggests to us the opposite. So we think these neurons are built to extended influence over very large parts of the brain. If they don't work properly, you really must start to think about ways to control that appropriately. So in a sense, we would go out there now and we would try and talk to clinicians, have a second look at this structure. The prediction is that if they are misfiring and spreading these bad rhythms across the brain, that if you can correct that surgically, you should have a relief from symptoms. And in fact, although it's not part of the larger sort of scientific accepted knowledge in this field, there are some tantalizing glimpses that if this structure is targeted in surgery for Parkinson's, it really provides remarkable benefit in terms of a relief from these movement difficulties in people with Parkinson's. So that's a first stage. This study, of course, was based only in Parkinson's animals, so we really don't know how these two different types of neurons, or at least this new type of neuron that we've discovered, we don't know how that cell type should be working normally in the brain. So if we then take our knowledge of disturbed function in Parkinson's and we compare and contrast that with what these cells should be doing normally, we can really get a better feel for what elements of their activity or other structural or molecular properties should we be targeting in treatments to provide new rationale for new treatments in Parkinson's. For example, if we're really thinking of sort of blue sky avenues for sort of therapeutic advances in this field, we might then say look at the molecular profiles of these neurons that we've discovered, and we could take advantage of these different molecular profiles in a way, for example, using gene therapy, you could express certain molecules that when activated will modulate the electrical activity of these neurons either up or down, calm them down, excite them. It's not as Star Wars science as it may have sounded 10 years ago, but one can imagine a day where a combination of gene therapy, expression of this and that molecule, combined with, for example, the implantation of fiber optics in the brain to deliver light in a precise temporal and spatial manner, to fine-tune and modulate the activity of these neurons that are quite frankly out of control in Parkinson's disease. Now, I think the kind of blanket approaches that typify surgical interventions in Parkinson's today, they can be very effective for individuals, there's no doubt about that. But I don't think any of us believe they're as good as they can be. So this is really where fundamental research of this nature comes to the fore. It points to this way and that way, and where we can selectively modify the activity of neurons, which we hope would lead to increased therapeutic benefit and perhaps fewer side effects. So that's where it could go in the next decade or so.